Well, good morning, everyone, to people in the room and to those who are joining online. It is my really great pleasure to introduce our speaker today, Dr. Jian-Min Yuan, who is a professor and associate director of the Cancer Control and Population Sciences at the University of Pittsburgh Hillman Cancer Center. He also holds endowed chair in cancer prevention. He has many leadership roles, co-leader of cancer epidemiology and prevention program at the Cancer Center. And his primary focus, many of you know him very well. So primary focus of Jian-Min's research is epidemiological studies, collaborating with colleagues who have expertise in biomarker development and application, and using it in the cohorts that he's leading, Shanghai cohort study and Singapore cohort, but also more recently, he is the PI of epidemiological lung cancer cohort in Pittsburgh, looking at individuals at elevated risk for lung cancer development, people who smoke and are at highest risk for lung cancer development. So I will not be talking too much about his area of research because he will give a presentation, but I just want to say that more than anything else, I'm really pleased to introduce Jian-Min because he's a wonderful colleague, really colleague and friend. He is a fellow Minnesotan. You can't become non-Minnesotan once you spend some time here at the university. So we are really happy to see you back, even for a very short time, and the stage is yours. Thank you.
Yeah. Thank you. Thank you. Very nice introduction. Forgive me, I have a little sort of infections as I share with Arena over the time. So I'm here because I don't know, these bugs are a little bit sticky, longer than I expected. So maybe bear with me. I don't think it's the infections because I already tested multiple times. So, hey, Steve, I just saw you. So today I'm very, very happy to be here because it is the place that actually I started from here. A lot of work today. I really try to put it together how the work here is greatly accomplished over the time when I was here, and I continue to work on it. It's not very, very novel to you, some of you, but it is the one actually can see the impact, the collaborations from the population scientists and from the laboratory, like Steve or the Arenas or Steve or here. You guys develop a wonderful, high technologies to really to sort of determine the very specific kind of exposure. That's the difficult part for the population scientists like me. So I don't want to, so here I just give you sort of very generic titles, but I look at 3 areas. I will give you examples how to move this. So I don't have any conflict of interest to declare, but basically I just focus on this, give a little bit background on the lung cancer's burdens. Certainly cancer still are biggest, cancer burdens, to our society. And then I sort of wrote a little bit on the tobacco background, how the tobacco and smoking and the lung cancer connected. And basically I will focus on the 3 areas. So one is, of course, how the exposures contributes to the variability, the variations we see it, and then the genetic accessibilities, how to contribute, and then also people exposed to the more protective factors, how we, really understand it to eventually contribute to individual risks on this one. So here, just give a very, it's a generic kind of, I'll give you, maybe most of you guys know it. The lung cancer is still the most common cancers. You can look at it, is number 2 in terms of cancer incidence, right behind the breast cancers. So you have about 2.2 million new cases every year globally as an estimate, right? So you can look at it on the lung cancer mortality. It's about 1.8 million lung cancer deaths every year. So these are number 1, almost count probably close to 20% of the total the cancer deaths. So that's like lung cancer still the big, in terms of the cancer field research, if we want to reduce the cancer incidence as well as to reduce the cancer mortality. So that's sort of the one you look at, in the US, you see that, although the incidence rate is ranked third after the breast and the prostate cancer, but in terms of mortality rates is almost identical as globally, right? Still 20% of the total cancer deaths in the United States. It is the lung cancer. So that's why a lot of research is still going on, So in terms of, So in terms of how the tobacco eventually linked with the smoking, this is almost like 70 years, right? So you can look at the early days. In the 1950s, the most earliest population-based study to link with tobacco smoking and cancer, lung cancer, is Richard Dawes and Ernest Windows, two classical case control studies. They link with this one. So after that, they initiate two large cohort studies, one in the UK, one in the US. So establish the tobacco smoking is underlying cause of lung cancer. So in 1964, so Surgeon General basically concluded that smoking is the cause of lung cancer so far. So then, of course, over the years, then a lot of policy developed to control the tobacco. Like in the 70s, of course, tobacco banned in the broadcast bans, and then later on in the late 1990s, so 48 states of the United States, settled with the tobacco, 4 large tobacco companies. Basically, they have to pay back for the health costs by the tobacco product. So then, so far I checked, is the tobacco company already paid $165 billion, from the 1999 until 2024. So it's huge money. They continue. I think most of the money is paid out, but they still have a smaller later settlement continue on this. So most tobacco control really impacts as in 2009 in terms of the US Congress passed a Family Smoking Protection and the Tobacco Control Act. So that gave the FDA the power to regulate nicotine products. So that's sort of, continue on this line of a lot of tobacco control work, and a lot of NCI even have a collaboration with FDA to create a joint program, try to control that nicotine product. So it's very funny because I always sometimes joke with Steve, even the nicotine is really not a carcinogen, but certainly as a politician or as all the people they recognize is the nicotine is the addictives. So they sort of are targeted on this area, but it's not targeted on really to reduce the cancer, but really reduce the addictions, try to weed out from the tobacco smoking. Over the years, so this all the anti or so-called try to smoking cessation campaigns. So over the years, you can see the tobacco prevalences go down dramatically. Like from the early 60s, almost like 50 or about 50s. So far now, we are around 15%, in this country. It's quite a lot of accomplishment. Now, the remaining 15 or 10% is very hardcore smokers. Now, it's very difficult. I mean, Steve, all of you in the rooms, as I work on this field, it's very difficult because a lot of factors behind this. They try to develop a new, different kind of smoking kind of device, the new products to see if they can help them to reduce it. But still almost like 15, 20, or 12 to 15% of the people continue to smoke. That is account a lot of impact on the health. Of course, if cigarette smoking goes down, so you can see the lung cancer risk also going down. So in both from this incidence as well as from the mortalities, although they have probably like 30 years behind the smoking curve, so you can see that clearly is reduced. So let me change a little bit. So of course, we all know smoking probably attribute about 80% of the lung cancer cases. Currently it's quite, sort of consistent data over the years. So on the other hand, so only about 1/4 of the male smokers over lifetime, they eventually will die from lung cancer by age of 85. For the female, have a little bit lower, probably like a 17, 18 kind of range. So then give you one, okay, yeah, 80% of the lung cancer can attribute it to smoking, that's good. So then on the other side, why is the remaining sort of like a 60, 70% of people, they are spared from the lung cancer? Can we learn from this? Something can help us to develop, smoking or cessation one way. On the others, can we learn something, can develop the strategies to reduce the lung cancer incidence? So that's sort of my really the thinking after this sort of, in terms of the variabilities contributes over here. So then let's look at tobacco. Of course, if we want to look at the variability on the disease, so first thing is you look at the underlying causes, right? The underlying cause is the tobacco carcinogens. So let's look at how the variabilities contributes to each or individual smoker's risk. So why they are different. So of course, they expose the tobacco carcinogens different, right? So then how to assess this one? So I just give you very rudimentary kind of definitions, for example. This is smoking on the middle, sort of in this column. I don't want to go to details. The earliest studies from the UK and from the US. The first definition is that define a smoker is you smoke 1 cigarette a day, continues for 1 year. So that you define as a smoker, right? So then over the years, in the US is a little bit slightly different. There's they're persistent smokers, half packets a day, then later on, in terms of the cohort studies that give the similar kind of definitions. What is the current definition for smoke over here? This is the most recent is American Cancer Society's definition for smoke. So they define is 100 cigarettes a day over lifetime. They smoke, okay? And they are continuous smoker now. So then you can compare, because we always want to assess the smoking with the risk. So you are comparing the smokers and non-smokers. So basically the question is on the non-smoking category, it's a lot of variabilities, right? So that's the one thing to keep in mind when you are reading the attribute to the smoking. So you have to be careful. So what are you talking about for the smoking, right? So each study, over the years, they change it. Like Shanghai, Singapore, we did it. We take the middle road. So they 1 cigarette per day for 6 months smokers. So we define as smokers. Because it's very difficult to define because people vary. So if I smoke 1 cigarette in my entire life, in the last year in the Christmas parties, after that, do you label me as a current smokers? Right? So that kind of things, it's in terms of humans, is variability. It's very difficult to quantify. So that's why in terms of smoking, self-reported data, you need to take a grain of salt. So when you interpret data, so of course, objective marker. So we are talking about objective marker is increased assessment of the exposure can establish a more clear, association or stronger. So here I just give you an example, right? So because tobacco cigarettes give you nicotine, that's how the smokers are continue to smoke. Because for craving for their nicotine. So nicotine, of course, come into the body, they're converted to the cotinins, and then eventually is all these cotinine byproducts coming to the urines. So can we measure them, right? Can we measure them? Here, actually developed Steve Hack's lab, and then Sharon Murphy's, the lab is really developed the world top notch in terms of this area of research. So when I was here, so to take this kind of opportunity, we have a population-based studies. We just collaborate to see if we can just use urine samples collected from the people in the free living world and send it to the lab to measure all these, we call it total nicotine metabolites, and to see if this can associate or can be assessed the variability of the cancer risk. So I switch gear a little bit to introduce the 2 cohort study I work on. So this is the first study, it started in 1986. I was in the late 20s. I just finished my medical schools, and I don't like the clinical settings. Really, I just every day. So I just don't know what to do. So I just switched to on the epidemiology side study. So I visited the USC at that time, as an opportunity. They said, oh, can we just create a long-term cohort study? Never done this before. Okay, never done this before. No one wants to do it because they even don't know how to implement. So I was young, of course, young people always more brave or more stupid to jump on it. So I just, okay, I will take it. So they're giving me $100,000. Okay, it's a lot of money. $100,000 only at that time. I was earning $5 a month. So they give me a lot of money. I just bring to Shanghai. I said, yeah, let's try to see if they can really works. So they took us 1 year. It's really, we can enroll a couple hundred people. We set up infrastructure. It looks like it's durable. So basically we interview the people, and then we collect the blood, we collect the urine samples. It's very difficult, of course, the freezers, we even don't have in China. I mean, in the 80s, we even don't have the freezers below -4 degrees. All the freezers, you need -20, -80, we have to import from the US. Takes a lot of effort, but the long and short is succeeded. So basically, after $100,000 spent, we coming back, ask NCI, can we start a cohort? NCI said, oh, these are very novel new things. So they agreed to fund us. So that's why they created the Shanghai cohort. So it took us 3.5 years to enroll about 18,000 men. We only on demand. At this current standard, no, God knows, NCI never get funded because you guys only look at men, right? You have to put it in women because our argument is, okay, men have much higher cancer incidence or cancer risk. So you can reduce sample size. If you put a woman, you double the sample size, you double the money. So NCI buy in and said, fine, that's eventually they agreed to pay for that. So after this 3 years for enrollment, then of course, we continue follow up until 2018. So this is the cohort study we will use a lot in terms of the, in the next few study I will describe it. So after finish this 1998, so I think Tiananmen Square, all this you guys know, last night in the dinner table, why I decided coming to the United States. So that's a trigger. So really to make me think about, should I stay the entire life in China because of this political turmoil? So I decided maybe I just take a few years to leave. Just take to look back, and then I will take also study a little bit more on the epi side. So I take a leave to the USC to work on the PhD degrees with Mimi. So at that time, Mimi started to think about Chinese health study. So then, of course, I learned quite a lot from the field. Then I just right away involved with Mimi to set up the Singapore Chinese health study. So that's how the Singapore Chinese health study started. So Mimi is my mentor. Mimi Yuan, she retired from here, I think it's about 15 years ago, I think it was. So it's time flies. So she started, basically similar kind of model as the Shanghai, but it's a little bit more sophisticated because of course we learned quite a bit and developed more comprehensive questionnaires. And then also, we are thinking, including women, of course, we cannot leave the woman out. So that's why, so eventually we got both. We enrolled 63,000 people from 1993 to 1998 to interview them. But it's just too much work because in the field, we cannot collect the blood urine samples. So we did not do it at the first phase. So we finished, we enrolled. So then in the second phase, in the renewal, we went to NCI. Now we are ready, we set up. So we can go back, these people, we can collect by specimen. So that's why NCI eventually funded. So we got so that's what the biospecimen cohort is a little bit smaller, but still have about 30,000 people. Okay, total, we have it. So we have urine, blood, all these samples are collected. We follow up until now. So continue follow up on these people. We used passive linkage analysis in the Singapore because it's a much better situation, not like a US people move around. The Singapore is the small island, people don't move out. So everybody, because government subsidized the health systems, if you want to go to hospital, you have to take your ID number out. So we have the ID number with us in the record, so we can link with all the cancer registries, all the medical conditions. A lot of these studies are continue doing this. So this setup was very well. So now coming back in terms of, Sharon Murphy's lab certainly measured all these total nicotine metabolites, you can simply look at this sort of, eventually we total nicotine equivalent over here, right? So we measure in the baseline urines, we measure the urine level of the total nicotine equivalent. We follow up for 15 years after the urine sample you collected. So you develop a lot of lung cancer cases. Now you just to divide the people at the baseline. These are all self-reported current smokers, of course, right, at the baseline. So then you can further separate them by the level of the total nicotine. So you can see the people who have a high level of baseline total nicotine levels, the lung cancer risk is about four times high than the people have lower. So these are very clearly demonstrate certainly people's smoking behavior different. They are inhale very different kind of nicotine levels. Right, so that's the one. So this is the Singapore, this is Shanghai cohort studies. So we replicate in the Shanghai cohort studies. It's almost identical for this, that. So these are, it's very clearly, it's sort of, tobacco smoking behavior, inhale, and the uptake of the objective markers can further increase in terms of the assessment. That's also explained some of the variability among the smokers in terms of their lung cancer risk. The second, of course, these are the slides I just summarized a couple, almost 20 years ago. So what's the carcinogens in the tobacco cigarettes, right? So, I listed, these are bigger categories. Total, I remember still, if I remember correctly, is 74, 76 kind of number of carcinogens, is established or proved in the animal models. So that's the numbers I remember. But I just focus on one thing, just a topic for today's topic, is nitrosamines, because these are tobacco-specific nitrosamines, NNK and NNN. So basically is more specific for the lung cancer development. So similar kind of study thinking. So now you have the nicotine, as I said, the nicotine is not really directly carcinogenic, but it is the nicotine comes in with the carcinogens, right? So that's the we are thinking, can we look at on the carcinogen side specifically, what does it look like? It's not that simple, okay? It's not every cigarette is made the same for the carcinogens. So here is the survey did by CDC a couple of years ago. So you can see the contents of the tobacco-specific nitrosamines in the cigarettes. So on the left side, this one is in the Shanghai market. The red color is the Marlboro, is the US brand, sell into Shanghai. And then in the China market. And on the left side is blue bar is the Shanghai or China local brand. So you can see the difference between sort of same cigarettes, people say, I smoke 1 or 2 cigarettes. They are exposed to the carcinogens, very, very different, correct? It just depends on their brands. So that's why when you just ask how many cigarettes you smoke, basically it's not really accurate to assess the lung cancer or carcinogen exposures, right? So here is in the China, and then here is in the India, is a lot of market similar kind of patterns. Somehow that's why in the US, I don't know, these smokers are exposed much more high level of carcinogens because of the tobacco they made this way, right? So then also we did, this actually, arenas sort of work a lot, in terms of this measurement. So we also collected 43 brands of cigarettes from the Shanghai market. So we actually focus on the people who report they smoke this brand in our Shanghai cohort studies. And then you can look at it to measure their contents of the tobacco-specific nitrosamines. It's even all local brand still are very different. The concept here is just, okay, all cigarettes made are not equal. So that's, you have to keep it. Sorry. So then we are talking about tobacco carcinogens. You have to think about, how to develop more objective markers to measure them. That's the thing, Steve Hack's lab certainly did a lot of work. And so that's the one, actually. They, work out the details. I'm not expert on this one, but I certainly can illustrate. So you're coming from the NNKs, goes to NNLs, metabolites. So eventually you can measure this called metabolites of NNKs. So then you measure this, and then on the individual basis, So the question is, if you have a high level of the NNKs in the body, through the urines, they also have a higher risk for lung cancer. So exactly this study we designed it, still has a rabbit run it. So basically, you use a baseline urine samples and then to quantify, so you can look at this as relatively smaller. We only about 100 cases early, so you can see about 3 times the risk of a high level of NNAL compared with the lowest totals. So these are, of course, all smokers. So then we extrapolate this, apply this same study design to this Shanghai cohort studies to give you is a much stronger association for the 3 times of the risk. So this to give you some idea, so how to assess in terms of epidemiology studies, use the human population to assess the variability on the risks, to apply the unique the biomarkers. That's why it's so critically important to develop very specific biomarkers can apply. Otherwise, I still can use the question how many cigarettes you smoke, try to analyze this data. You just cannot move forward, right? So this is the really, I think it's the unique in here developed specifically. So I will switch to the genetic. I'm not personally not really work on this a lot because I really want to give examples of how this genetic thing works. The genes certainly contribute for the risk. So give you, sort of thinking about why this way, because gene can modify smoking behavior, right? So then, of course, you change your smoking behavior, of course, the smoking leads to the cancer, lung cancer variability. Some genes, we're still not very clear, maybe direct impact on the sort of cellular kind of functions, some genes. It's not a whole lot. So then in the past, probably 15 years, earliest GWAS studies actually is reported by Chris Amos. This is the summary, is by Chris Amos's the group, now in the Baylor. So you can look at over the years. The earliest study is actually in 2008. 3 GWAS published at the same time. They identified 3 areas of the loci in the genes is 15q25, and then 5q15, and then 6p21. So basically, is the earliest I listed these findings over here, right? So you can see this, the 15q25. This is the nicotine, acetylcholine receptors. So basically still are modified the nicotine neural effect. So basically, people carry that genes or mutations, their smoking behavior different from the people who don't. So that's kind of things, this is the very significant p-values, and this is the odds ratios associations, right? The similar correspondence. So then also you can look at the other genes. This so-called overall, you can see all the other SNPs or loci 45, besides these 3, it's very low. It's very relatively, it's associated with the disease risk almost like in the 1, almost in the 1. So that means the effect is just not there genetically. So in terms of how much really to contribute to the lung cancer risk, besides the smoking or through the smoking pathways, it's minimal. It's minimal so far. So I continue to work on it. I don't think they have get, because after that they did, I think these slides they put up, I remember the 40 GWAS in the European study populations, 24 GWAS in the Asians. So how many GWAS you want more to study? So I think that's the way, it's just as much as you can do. They just cannot find new things anymore. Right? So that's the way I think I look at the genes, okay? Now I put over here also this one, it's here actually, is the one genes is actually quite interesting in terms of modifies a lot of nicotines. Here's Heather Nielsen with Kinney's labs, basically we developed a device to focus on this gene. The GWAS cannot pick up because the gene structure is very, very complex. They have deletions, they're shifted. So all the GWAS never found out. This pick up where is actually OncoArray. It's not traditional GWAS. They develop OncoArray. They know this gene there. They put a tag snippet there. It's very minor. They find it. It's a relative risk of almost 1, almost nothing. So that means it. Just compare the later. I can give you the stories. So basically, GWAS not very succeed in terms of the functional genes you are looking at. Just keep in mind, it is important work. Okay. Personally, I'm not working on this, but it is the one. The whole field are moving this forward. So just look at the focus on the CYP26. It's very polymorphic. So far, probably they already have 70 kind of variations identified in the various populations. You can look at these, alleles variations. Each different kind of population have very different kind of frequencies over there. So in the Chinese population, that's the area. So we thought we can do something about in terms of use the genotype work we can do. So that basically, as I said, is the same thing I alluded earlier. So that the primary function for the cytochrome P450 2A6 is the primary enzyme to metabolize nicotine, to convert to cotinine, and the most important cotinine to 3-hydroxycotinine. So they basically, you can genotype them. You also can phenotype them. You can check their function, because there's almost 90% of this nicotine goes through that pathway. So if that gene is dysfunctional, so you can see the difference. Basically, it's a Sharon Murphy's work, and if you still has, also you can see that measure the nicotine, sort of the ratios, right? And we call the nicotine metabolizer ratios. Basically, you use this cotinine or the 3-hydroxycotinine versus cotinines, basically assess the CYP26 functions. Of course, on another side is maybe this CYP26 or similar kind of class of the enzymes may contribute to the activation of the ANK. We don't know exactly. We still have a lot of work. Maybe still you can comment on this. Is there any development on these areas in terms of how this cytochrome P450 2A6 in terms of metabolism on the ANKs. So we did this genotype work, use both Singapore and Shanghai Corridor study. We have the blood samples. We take the DNAs. We in Heather's lab and also running in the Kenneth course. So we did quantify in terms of allele frequencies on the CYP26. So basically focus on these 3 or 4 allele variants. So it's functionally, we did not do the function work, is previously already published. So if people carry these star 1s, actually they decrease their function, decrease. If people carry the star 7 or star 4, they lost function. They don't have the function for this one. Or the people have the star 9, so they basically is actually decrease the functions. So that is the ones you can create the combinations of these genotypes and then can assess their functional, okay? So here is the function. So we did also measure the urinary so-called nicotine metabolizer ratios, and you line up people by these allele variants for the combinations. So this is a normal function, right? So the ratio is 2.26, the ratios. So that means it's normal. And if people who carry the loss of function, so they're only 0.18, 10 times difference. So then it's these genes, whatever the genes, we quantify over here, and it is functional. It's very functional. So then we just group them because sample size is small, so we group as 4 categories, right? So after group the 4 categories, you can see is if the people who carry the poor genotype, so they only have a 1/3 of the function, then the people have the normal function. So it's dramatic. It's very significant. Now it's okay. People have lower these functions. What is their consequence in terms of this one, right? So what is their changes in terms of behaviors? So you can look at it. We also replicated this in the Shanghai, in the Singapore course study, almost identical. It's identical with the results. So it's very clear. So then you can look at also you can look at the uptake for their nicotine. The people who carry the lower or no functions, So their total nicotine equivalent much lower. That means they don't smoke as much, as intense as the people who carry the normal functions. So that is also contributes to smoking behavior. Of course, that leads to the reduced level of the carcinogens. So you can see the carcinogen level also quite lower, about 40% lower. So there it's very simple. You can translate to the lung cancer risk. Like the people who carry the poor genes, they smoke less, they have less carcinogen exposure, so have a lower lung cancer risk. So then you can look and compare, is only quarter of the risk of lung cancer if they carry that loss of function cycle 6 to 6. It's 4 times. Yeah. So you compare the early, you remember in the GWAS, you barely see anything. Right? Because they just globally try to quantify, just not accurate, just not accurate. So this is the one. So this is just debatable, I just still put up there. So after we adjust all the tobacco smokings, all the behaviors, there's still, if the people who have sort of, the poor genotypes, they still have a lower risk. So the question here is I cannot answer, maybe still you can, what else behind this? Right? Is the genes play some other roles, or we are not completely quantify these exposures enough to really to even this kind of take this account for these risks. Okay, so now I think, what's my time here? Okay, I have about 20 minutes or maybe 15 minutes. I think I can run through. So now I just switched the gear to look at on the protective side. A lot of protective work, you guys maybe remember, in the early days, antioxidants, beta carotenes, vitamin E's, all these, things are worked out. MCI has put a lot of money to the clinical trials, did not work out. Just totally failed. People who put on the beta carotene, they have 20%, 15% increase in lung cancer mortality. Totally failed. So this field still have a lot of rooms to fill it. So here is actually, I picked this isocyanate because of course, the part of the work is from Steve here, and I continue interest on this area, can work, so sort of work on the crucible vegetables, certainly because I have a lot of mechanism, animal models to work out behind, not like a beta carotene. They don't have any, laboratory data, any mechanism study, they just jump on it to do the phase 3 clinical trial. Just doesn't work out, right? Just doesn't work out. So this just give you if you're not familiar, that you maybe can find it from, very common in a lot of these in the grocery shops, around here. So basically, I think our really interest on this one is watercress is one, and as well as in the sort of broccoli sprouts. I mean, in terms of the work we do it. So here is a, sorry. Is a meta-analysis did by IAC group, sort of try to summarize all this consumption of the crossover vegetables to see if these are traditional epi studies. Just ask, do you eat and how much you eat in crossover vegetables, how frequently you eat. So basically, conclusion is if the people who said they eat more frequent crossover vegetables, they probably have about like a 15% kind of lower risk of lung cancer on global scale. This is the cohort studies and this case control study. So around that ballpark. So that is the sort of the epi studies I've summarized how this eventually these vegetables with the lung cancer risks. Now, two things I want to point out is, of course, in each crossover vegetables, they give you very different kind of isocyanate. Steve Haxel work really focus a lot on the watercress, eventually leads to the sort of produce the PITCs. So this, the agents that you can use, and also use broccoli sprouts, eventually goes to the sulforaphane. So that's the two areas, two studies I want to describe. So of course, the isocyanate, ITC already identified, in the laboratories, a lot of basic work to support and have chemopreventive properties. So then we try to apply this to our population studies. So that here is actually first thing we ever did is apply the urinary markers. These are markers applied to the Shanghai cohort study. Similar designs, have a baseline urine samples, and then we take the urine samples out and send these urine samples to the lab to measure the presence of the total ITCs. This after 10 years later, so we just ask the question, if the people who have high level of the presence of the ITC in the urine at the baseline, are their lung cancer risks increased or decreased? So this is the one to show. So if the people who have the positive urinary ITC in the baseline urines, and they reduce about 35% of the lung cancer risk in the Shanghai cohort study. So that's the sort of the trigger. This is the first sort of a little bit on the more objective, rather than use the self-reported questionnaire. So these are more objective kind of markers linked with the lung cancer risk. So of course, this one, we also have debate on this, if their GST-M1, GST-T1 modifies the risk. Now for this study particularly, we see a little bit stronger in terms of the reduced risk for the isocyanase presence among the people who carry the no genotypes. So that's sort of the original kind of epidemiology study reported. But later on, some, the data that's not consistent or find it similar, but I just want to tell you what we found early. So based on these data, of course, Steve Hack here, all the, a lot of here actually, we carry out a clinical trial. It's just give you very briefly. So basically is the use PITC is the extract from watercress. So basically try to assess if PITC will modify the NNK metabolism, because you remember NNK is really the prime, the lung carcinogens. If we modify, so by definition, maybe can reduce the risk of lung cancer. Here, just basically the M's. So the modify NNKs, and then of course, also to modify all the increased excretion for the other environmental or tobacco carcinogens, as well as look at the genes, GST-M1, T1 is modify that multipolisms. So just to give you a little background, so NNK comes in as a metabolism, coming as I alluded before. So you need a step of called activations. So eventually leads to the activated compounds that attack the DNAs to damage the DNA cells, eventually leads to the lung cancer. That's very simplified schemes to think about for this one. So you can measure the urinary, this keto or this I call it hydroxy or keto acids as the active compounds to see them. So because of a lot of nicotines, some nicotine can influence impacts on these hydroxys, a very trace amount. So the original kind of design is very difficult to quantify correctly. So that's why the Steve and also Arena, sort of the labs that generate, so add this kind of deodorant in case to the cigarettes. So then basically you use a tracer. So you look at the tracers, eventually can measure the tracers, and you can actively quantify the sort of the metabolic pathways from here. So then here, of course, the hypothesis is the PITC maybe modify that genes, the enzymes. So it can modify this eventually the ratios of these activations. So this is the measurements, right? So we are measuring all this sort of from this, D4, hydroxy acids, and then to ratios to see if the ratios are high, so that means more activations, pathway. If lower, so that means it's suppress the pathways. Okay, the study is actually, it's a simple design. It's very difficult to do it because I know we need to collect the urine sample. I remember we need to collect like 2 weeks, 24-hour urines, like, 5 days, to carry around. It's a very, very difficult subject to participate, to complete the study. But, we managed to do it. So these are crossover designs. We randomized 82 smokers. They first they give the PITC or give placebos. And then after 1 week, 5 days treatment, and then after 1 week washout period, they just cross over to give it. It's very simple, straightforward design because they can control by themselves, and then to resist the results after how many years? 3-4 years of exercise. Just a lot of work. So this is the main results. Funny, the people who put on into PITCs, they have about 7.7% lower so-called, activations of the NK. It's there, significant, but the magnitude of the effective size just as low as we expected. So that's sort of, put over here. That's sort of so sometimes that's why you jump into the studies, you don't know what you're getting, right? So after a lot of these exercises. So we also look at, GSTM1, T1 is not really modified that much. So that's sort of, not really modifies. It's not there. So here actually, I just point out for another compound is benzene, right? It's very interesting. So benzene, certainly here is benzene, how the benzene metabolites, eventually you can measure SPMA, so is mecabic acid. So you can assess the excretion of the benzene through the urines. So the hypothesis is PITC may can modify the GSTs because the GST is critical for the benzene metabolites to the mecabic acid pathways. So you can see this sort of. So then we just measure the participants' urines before and after treatment. So overall, the people on the PITC arm, they can increase 20.5% excretion of the benzene. I think it's substantial, sizable. It's interesting, particularly for these high exposure people because these are high smokers. So that's why it's clearly demonstrated. Also very interesting is actually GST-T1. People who carry know the impact of the PITC to increase the excretion of the benzenes through the urine is much stronger. So you can percentage-wise, so you have increased 75%, right, for the benzene excretions. So then you look at the GST-M1, also have a little bit. This all is on the borderline significant, and then you combine these together, both no genotype carriers, so they have about almost double their benzene excretions. It's a very unique kind of data, very, very interesting, sort of, this kind of even small scale study, so you still can demonstrate so that kind of work. Now, I will not go into detail for the other three. So other three is actually the acrolein and the 1,2-butadiene as well as chloroaldehyde. They all increase. You can see this is the black dot is the overall. So in the PITC treatment arm, then all the other three is genotypes. So it looks like they all have a little bit increase, but not as strong as here is actually is the benzenes, and here is the NNL metabolism changes. So this work looks very promising, interesting. So then, Steve Hack continue to work on this, but I also interested on this area. That's why we develop another trial. So these are very new data and have not published yet, but I just want to share with you guys what we find. So we develop because our PITC is very difficult, as still you remember. So we used olive oil because we cannot make a pill. So put an olive oil, put a syringe, ask the people squirt in the mouth. It's just as nasty as you can imagine, okay? The PITC itself is very small.
Cost $5 million.
Yeah, because we tried, I still had to walk around, we went to tour a few facilities, just cannot make it. So that's why we a little bit sort of here, because we choose the sulforaphane, so the pill is available. That's why sort of we decided to take this route to see if we can develop and finish the study. So basically very simple. The idea is that we give the subjects sort of, these are former smokers we focus on. So we have a little bit long term, we are trying to do 12 months, the treatment. And we also want to assess the histology changes in the lung. So we use the biopsy samples at the baseline and also after 12 months. So we are follow up the subjects for a year at least. So the basically two primary aims is histology changes as well as for the cell proliferation and apoptosis biomarkers to see if we can demonstrate some kind of changes. And then also we look at some toxicities as a gene expression profiles. So I don't have the data for the gene profiles, but we have just finished the analysis, so I cannot show, but I can show the first primary aim. So basically, So we are focused on the smokers who have a history is heavy smokers. Say all the damages are lung, so we have CT scans at the baselines, and they have lose their lung function already, some part of it. And then also they don't have the disease, the cancer at the baseline. We screen them out, and then we put them to recruit them. So basically it's 1:1 ratio randomizations. So we use the pills, I think it's of 8 Amercol. The pills each is 15 milligrams. So eventually converted to about 120 micromoles of the sulforaphane. And then also the company made the placebo pills for us. It looks identical, but not discernible by the study subjects. So then basically that's the outcome we are looking for. So eventually that's the treatment. I just described how we develop treatment. It's basically 120 milligrams of tablets, and then converted to about, is 15, I think it's 120 micromoles of the sulforaphane. And then here is the lung tissues, how we collected. So we predetermined 6 sites in the lungs. So this is the sort of, in terms of the 6 sites is a light, up, lobe, then also the right, up, these are all the pulmonologists decide. So we have to pick it more safely. So this is the histology, codings. We of course exclude these 6, 7 at the baseline, but later on we just include at the end of the study. In terms of cellular kind of proliferation markers, here we are focused on Ki67 as well as the Caspase-3 and the tunnels. So that's the markers that we are looking at the tissues at the baseline as well as after treatment. So then the monitor adverse events, of course, every 3 months they come in, self-reported. We also test the liver functions, of the 12 months of treatment. So we did not find any severe, toxicities. So this is the numbers. Originally, we planned to have 80 people to be randomized because of COVID in the middle, territory shut down. So we only got about 43 people we randomized. So some people drop out. So we finished about 20 on the placebo and then 17 in the treatment group. It's a little bit smaller sample size, but I still see some signals. It's interesting. This is just descriptive data. The history of smoking are very comparable. It's very heavy smokers. You can see their packages is almost like a 50, 40 heavy smokers. And then also you can look at this, there are no side effects at all. So we see nothing. It's really just a normal kind of GI complaints from the people, even in the placebo groups. So we did not find any changes in the histology grade after 12 months. We did not change, adverse or progress or regress in terms of histology grade. Not very much. Now we indeed find that Ki67 is actually significantly different between the 2 groups. So this is the treatment group. This is the placebos. So it's a surprise to me, but that is the data. Just short, because these are all double-blind. It's all different pathology departments that generate data. So then if we further look at more intensity on the Ki67, because in the pathology report, so they can look at intensity 1, intensity 2, intensity 3, into the different kind of level of the Ki67. So it's actually much stronger in the high intensity categories to the reductions of this. So that is the data. We just find this, the whole 6 years work. So that's only the results we find. So It's interesting, now we don't know what to do. They also, we analyze these people compliances. Because people are first 6 months are very good, the drug. So later on, they start not complying, actually. So why we not? Because they reported to you, they always, okay, take it, always 100%. That we have pill counts, coming back, But we use urine sample to test them. So we collect the urine samples. So that's a urine sample. You can urinary level if are high for the sulforaphane, so their impact on the reduction of Ki67 is stronger. It's very clear. It's very clear in terms of if they are not very compliant, the effect is just not there. So then certainly it's from another angle is, of course, we have to do very diligent work, try to encourage people to comply. With the COVID, certainly we cannot have a very in-person kind of engagement. So we call them, we mail, we mail the drug, So that also maybe a little bit kind of, the loser. So that's, I think I have today, I have still a few minutes. Summarize sort of how this precision measurements of the exposure is critically important. Okay, I think in terms of to assess the lung cancer risk, as I give you the, nicotine metabolites, NNK metabolites, that is critically important. And also the genes, I think still there still room for the candidate gene approach if we really work out. So currently we have a grant with Phil Lazarus. So he collected a lot of lung tissue samples. He's trying to work on this metabolites with NNKs, and then try to identify is any kind of SNPs can relate to the lung tissues. Then we go backwards. So we try to identify candidate kind of genes first. Then we target on this area to continue to work on it. So then, of course, also the isocyanate, to me, I think it still have a lot of work to be done, but it's certainly, it's an interesting area. I hope the younger generations really can pick it up and continue to work on this. So this is the one. Of course, before I close, there's really a lot of work, a lot of work is here, right? Is conducted, is initiated here, is completed here. It's really thank all of you guys over here. There's a lot of people on the online. So thank you. It's a very, for me, it's very career-wise, it's a very, very fruitful journey for me to reach this point. Thank you all.
I have microphones for anyone with a question.
Yes, here. Question.
Oh, hang on.
That was a great talk. Thank you.
Thank you.
So my question is, and I should know it, but so when you have CYP2A6 deficiency, right? So you will have less metabolism of nicotine, so you need to smoke less, right? But wouldn't that also affect metabolism of NNK and other tobacco-specific nitrosamines? So how does that affect actually metabolic activation? And is the change in risk you see is due to decreased smoking or altered metabolism?
I think from my understanding from the slides I showed, I don't know, it's still what you agree. They have some additional function on the CYP2A6 we don't understand. From the surface, that slides I can go back to see if I can find out this one. Sorry. Almost there. So this is the slide. So you adjust for whatever we have measured, like tobacco intake, or the nicotine. They still have reduced the risk of lung cancer for the people who carry the poor genotype. So you can interpret this maybe to reduce the activations. That's why they reduce lung cancer. That's one interpretation, right? It's not necessarily covers all of them, but at least some of them maybe. So that's why we don't know exactly. No, I don't know how many animal models, cell line studies you can really demonstrate on this. Use this. Still, you can maybe comment on this.
I think the effect on nicotine more important in this case than the effect on
Okay.
the metabolizers.
Yeah, that's for sure. That's for sure. Yes, that's for sure. Because I mean, the things, because these are the black bars certainly on the nicotine side, right? So the
This is not really that big.
Steve, do you mind if I give you a microphone? It's just our virtual group doesn't get to.
So at least maybe partially address your question about maybe it is have another side of the function for the site, the 26 maybe on the activation side of the case.
I have one question from our virtual group. Clement Wilcox asked, do you see that these results would be applicable to people who are vaping?
Oh, that's a very good question. Now let's talk about the vaping. So first is what is the vaping? I always go to the kind of the bottom. The vaping, of course, is not typical cigarettes. The content is very different. They don't have much high level of NK there. It's very trace if there are any. I just don't think they have any present. Of course, they have other potential carcinogens, like maldehyde, the other organic compounds. So you apply this one, maybe you can apply on the nicotine side. Yes, you can apply to assess the nicotine levels, nicotine metabolism, and the genes modify the nicotines, but it's not directly applied for the tobacco nitrosamine. That's totally different. Okay.
All right, thank you. If we don't have any other questions, let's say thank you one more time for a great talk.
Thank you.